We're years into the future, and the world is grappling with a new Pathogen X pandemic. Tens of thousands of people have already been infected, with millions more expected to follow. You need to quickly and reliably test people for infection. To do so, you turn to qPCR, the gold standard in pathogen detection. But what exactly is qPCR, and how does it work? By the end of this video, you'll be able to 1. Describe how qPCR works on a molecular level for pathogen detection. 2. Recognize the key components of a qPCR. And 3. Learn about the quantification step of qPCR and what it tells us in diagnostics. What is qPCR? qPCR stands for Quantitative Polymerase Chain Reaction. It's a molecular technique that amplifies specific regions of a pathogen's genome and measures the rise in fluorescence as the target is amplified. This allows us to detect even a small amount of a pathogen in a sample. To run a qPCR test, several components are required. First, we need a clinical sample to test for the presence of a particular pathogen. If the pathogen is present, qPCR detects and amplifies a specific RNA or DNA sequence unique to the pathogen's genome. We call this specific sequence the target. In addition to the target, the qPCR contains several key reagents. Primers are short, single-stranded DNA sequences complementary to the target RNA or DNA region of interest. They bind to the target region we want to amplify. dNTPs are the building blocks for DNA, known as nucleotides. Enzymes such as DNA polymerase, which catalyzes the formation of the new DNA strand, is included in all qPCR reactions. If the target is RNA, you will need to include an additional enzyme, called reverse transcriptase, to first convert the RNA genome to complementary DNA, or cDNA. This is known as RT-qPCR. Buffer refers to the chemical solution that maintains the optimal conditions for the enzymes to function. All of these reagents are also used in traditional PCR, but qPCR has one more critical reagent that gives it its quantitative nature, the fluorescent probe. These probes are short DNA sequences that bind to the target region between the two primers. As amplification occurs, the probe is cleaved, or displaced, releasing a fluorescent signal that increases with each PCR cycle. Finally, once the reagents have been combined, the reaction is carried out in a qPCR machine, an instrument that combines a thermocycler, which precisely changes temperatures to drive the reaction, and a camera that captures the fluorescent signal in real time. Now that we know the components of qPCR, let's review the general steps to detect a pathogen in a sample. The process begins with sample collection, obtaining a clinical specimen to test from bodily fluids. We next process the sample by performing a nucleic acid extraction, isolating the DNA or RNA inside of the sample. For RNA targets, we must next perform reverse transcription to convert RNA into complementary DNA, or cDNA, as the input for amplification. For DNA targets, you do not need this step since you already have your input DNA. To run the qPCR reaction, a thermocycler is key since it has the capacity to change temperatures over specific intervals of time, which is the foundation for the 3 steps of a qPCR cycle, just as in PCR. First, we raise the temperature to denature or separate the 2 DNA strands. Next, we lower the temperature to allow for annealing or binding of the primers to the target DNA strand. And finally, we raise the temperature again slightly to the optimal working temperature of the DNA polymerase for elongation, when the building of the new DNA strand occurs. As new copies of DNA are made each PCR cycle, the fluorescent probe first binds to and then is displaced from the DNA strand, emitting a signal. Over multiple cycles, the fluorescent signal increases as more and more copies are produced. The qPCR machine measures this rise in fluorescence in real time, cycle by cycle. Finally, the last step is the detection of the target. If the target sequence is present in the sample, fluorescence increases and eventually crosses a predefined threshold, indicating a positive result. If fluorescence remains flat, the test is negative. For positive samples, the precise fluorescent readout is key. It's how we estimate the amount of pathogen in the original sample, known as the pathogen load. To calculate this, we determine the number of PCR cycles needed to amplify a target to a point where the fluorescence rises above a set threshold. This is known as the cycle threshold, or CT value. In theory, the amount of DNA doubles with each cycle, so fewer cycles means more starting material. That means a lower CT value indicates a higher pathogen load, while in comparison, a higher CT value means there was less pathogen to begin with. Let's take a closer look with an example. Here are 3 samples containing the same pathogen, but at different concentrations. As we run them through qPCR, you'll see how the CT value shifts depending on the amount of pathogen in each sample. As you can see, the more pathogen in the sample, the fewer cycles needed to detect it. To turn CT values into actual concentrations, we use a standard curve, a reference created by running samples with known pathogen amounts. This lets us translate CT into meaningful numbers. With this information, we can determine whether a pathogen of interest is present in the sample, how much of it is present, and use that to guide diagnosis and care. Let's recap. qPCR is a powerful tool used to detect pathogens in clinical samples. Key components of the reaction include the sample itself, primers, dNTPs, enzymes, buffer, and fluorescent probes. Fluorescence is measured over the course of the qPCR cycles, and a rise in fluorescence indicates the presence of the pathogen. The CT value helps us estimate how much of the target pathogen was in the original sample. A low CT value indicates a high concentration of the pathogen, while a high CT value indicates a low concentration or possibly no pathogen at all. We hope this video helped you better understand how qPCR works and why it's such an important tool for pathogen detection. To learn more about Outbreak Response, check out our Outbreak Response video series. Special thanks to our dedicated team of researchers and the Broad Institute video production team, who supported the creation of this video.